Hello, everyone. This is Dr. Kajuri, an early career nephrologist and renal transplant physician from Hyderabad, India. I am a member of the social media team of the Winicon 2024. Today, I am extremely delighted and elated to invite Dr. Priya Alexander in this meeting. And this is one of the meeting of the pre-Congress interview series of the Winicon 2024 conference. Dr. Priya Alexander is a clinician par excellence. She is a renal pathologist with special interest in glomerular diseases in Mayo Clinic. Her clinical focus includes expertise in glomerular pathology, especially immune complex glomerular diseases, renal allograft pathology, pathology of the aging kidney, as well as thrombotic microangiopathies. Except besides this clinician activities, she is an educator and she has mentored many residents and students. She has also participated and authored in high-impact clinical journals. Her research is mainly based on understanding the molecular basis of renal diseases with the intent of biomarker discovery and renal classification. Today we invite Dr. Priya. Hello, madam, and as a part of this interview series.
Thank you so much for your kind invitation.
Yes, ma'am. Ma'am, we'll start the interview, and the first question will be, what actually led you to renal pathology, ma'am? If you can share your experiences.
Absolutely. So I trained, my undergraduate and postgraduate medical training was in Christian Medical College and Hospital, Vellore. And I was taught by excellent pathologists. And I can still remember sitting in the classroom in the Norman Auditorium and listening to Dr. Anand Date. Now, to those who know Dr. Anand Date, he was like one of the fathers of Indian renal pathology. And while I was sitting there, he brought the very complex topic of renal pathology to life. And his explanations were wonderful. And that was the first introduction to renal pathology. And then I started reading Robbins, and I could see the marvelous description of the kidney through the literature that was in Robbins' basic pathology. So I did not know then that I would pursue renal pathology as a passion, but it certainly began there. And then I moved on to work with excellent clinical teams in both in my undergraduate, postgraduate, and as a faculty briefly when I was in St. John's, and I worked with Dr. Isha Garg and Dr. Urmila Anand and several of these leading women scientists. Then I pursued my passion and interest with my family's interest to pursue studies abroad, and I had the incredible opportunity to train with a brilliant renal pathologist. I'd say one of the most brightest minds in kidney pathology was Dr. Helmut Renke at the Brigham and Women's, and he himself had been trained by Dr. Kotran. So it was a real fortune for myself. My interests were fueled. They were like seeds that fell in the right soil, you could say. And then I had the most amazing opportunity to join a Mayo Clinic, which to me is like, has always been an unimaginable dream to be here. This is a phenomenal institution. And what is so beautiful about it is that there's excellent pathology here, there's excellent clinical teams, and it's this combination of working with people who are trying to understand and solve diseases with a background of having resources like large amounts of data that can be accurately archived and collected, and then the ability to use resources that you have to translate them into scientific ideas. If we're lucky, you get great results, and then they can be results that can be utilized everywhere. So that is, in a nutshell, my passion and love for renal pathology.
Great, ma'am. Nice to know. And my second question will be, I have seen that you have a special interest in molecular kidney pathology. Can you tell us briefly, like what inspired you this molecular kidney pathology?
So a lot of my understanding of pathology in the kidney was driven by both Dr. Date, Anand Date, and Dr. Helmut Renke. Now, Dr. Renke really taught me to think. I would say many people are worried and fearful when they think of the kidneys. I think it's actually a remarkable mystery to be solved. And I think a lot of my interest in understanding the pathophysiological basis of renal disease stemmed from his insights as we studied cases. And so you can think of the kidney as being this amazing opportunity to actually apply immunological basis of disease, understanding monoclonal gammopathies, understanding infections, and in the transplant, it's a really great platform or disease set to study so many aspects of pathology. And so my first actual, I would say, real opportunity came when I was at Mayo Clinic working with this incredible team of pathologists. We are now 10 of us. Everyone is, I would say, just brilliant. And I started working a lot in with fibrillary glomerulonephritis with Dr. Sami Nasser. And previously, I'd worked in the area with some amyloid with Dr. Sanjeev Sethi and Dr. Favenza. And then these areas of studying proteomics gives you the opportunity to understand the actual proteins that are contributing to some of the big diseases that are at that time, when you don't know, and you have the opportunity with excellent mass spectrometry techniques here at Mayo Clinic. We're one of the largest centers, and it's a big referral center, and we have a phenomenal team here. So you can see periodically that I'm talking about being a member of a team, right? And that's always, you're not an island on your own. You work with a group. And so we were fortunate to be able to discover DNA JB9 through this collaboration with hematopathology, the mass spectrometry group, and my colleagues at renal pathology here. And we discovered that, in fact, fibrillary glomerulonephritis has a large, proteomic structure that has an antigen that's unique to fibrillary glomerulonephritis. I would say that was the big chance for me to understand the potential we have. And now you can do DNA JB9 stains in any lab at a very reasonable cost in any country. You don't have to wait for EM. I think that was probably my big motivator. Then there are now novel techniques that are further available, which are the single cell space. And this is, there's a lot of work that's been done in transcriptomics and other molecular tools where you don't have spatial orientation. The chance that I've had is incredible, where I'm now able to look at multiplexed marking of immunohistochemical based marking of tissues in a spatial context. So you can imagine where you can understand not just one marker, but 40 markers, how they label tissues. You can understand immune interactions. You can understand immune epithelial interactions. And this has had a big role to play in tumor. But now our work has, with my colleague here at Mayo Clinic, a lot of the work is now involved in looking at transplant pathology. How does it play in different patterns of rejection? We've done work even in COVID-19 disease when there was a lot of mystery as to how is the kidney being involved in COVID-19. And we actually showed using this molecular tool that you can understand that right in the kidney, there's both an innate and an adaptive immune environment. That these are the ways that we have used this technique. We plan to use this technique. And I'm sure in time we'll be able to do large collaborative studies that allow us to work with other countries and other keen clinical scientists who would like to explore unsolved issues. And technology is always going to increase. There's going to be several opportunities, but you really need to work with people who want to solve a problem and make it easier for the patient. And final outcome is, how do you improve patient care? That's my understanding, my approach to this. And I really believe that we should take the science out so that more people are able to use it, work together, and help solve patient issues.
Yes, that was a great thing, because this DNA JP9 has really revolutionized, and it is very easily available. We also do it all the time. So this leads to the third question, like what according to you, ma'am, are the emerging areas of nephropathology?
I think there are several aspects of pathology that are untapped and are in their evolving stage. You can see with the role of understanding, moving away from patterns of disease. Like for a long time, we called everything membranoproliferative glomerulonephritis, membranous. And you can see that all of this has changed, right? Membranoproliferative glomerulonephritis now is recognized as only a pattern of injury that can be seen in so many others. Now we're coming to disease-specific terms, and I would say that initial ideas of this, Dr. Renke had already sowed in our mind, and then you've seen the literature with Dr. Sethi and Dr. Favenza have really pushed the idea of how membranoproliferative glomerulonephritis can be complement mediated, and how this is what I think is the future, where you move away from patterns of injury to actual antigen or actual personalized understanding of what is going on with the patient. Is that pattern of injury from thrombotic microangiopathy? If so, is it complement mediated? Is it derived from an underlying monoclonal gammopathy? And the breakthrough with Dr. Ween's study in primary podocytopathies, that's groundbreaking, right? And that she always gives great weightage to the finding of Dr. Helmut Renke again of the podocyte dusting. So it really comes back to a simple observation that you don't dismiss. We shouldn't dismiss simple observations. If you take the podocyte dusting, it was Dr. Renke who looked at it and said, this means there's probably an antibody out there. And then it takes a brilliant mind like Dr. Weens and the right team to work together and collaborate with others. See, again, it was collaboration. We were just part of a collaborating team, but she was the mind behind it that had the ability to say, let's look at these podocyte antigens and see if we have an antibody. And look at that now. You have a group of, all of this is where I think we're going. We're going towards a personalized form of evaluating renal disease. And right now at Mayo Clinic, we've used all the membranous antigens that Dr. Sethi has discovered with the PLA2R. That was the groundbreaking discovery. And now we have, using mass spectrometry, we have a clinical test. So I think it's all about being, yes, there's always an element of being fortunate to be where you are. But then if you're so fortunate to be where you are, then you must also use your opportunity for good so that many are benefited, and working together and being humble, because we make so many mistakes, but there's also great good that comes out of discovery. Not every research idea is successful, and one has to take that and accept that. And I'm most interested in looking at transplant pathology with the molecular tools, because antibody-mediated rejection is a huge challenge still. And I hope to be investing time in these areas, using these tools that we have to understand immune landscapes that can modify the way we look at disease, and whether immune signatures of different diseases can be targeted. So that's what I hope to be looking at and covering in the talks that lie ahead.
It's great area, madam, human signature. And coming to the last question, like in the upcoming Winicon 2024, you have a special session on the talk about biomarkers. So can you give a small glimpse on your talk, please?
I think what I've covered in this interview is what I'm going to be talking about. And, it's essentially going to be this in a nutshell, talking about different successful stories, and then probably moving to what I'm most interested in, which is imaging, mass cytometry, which is the opportunity to study single cells in a spatial context. And I will talk about that in relation to transplant and native disease. And I look forward to the interactions with all of you. And I'm sure we'll all have wonderful ideas to work together in the future and challenges that we can work together to overcome.
Great, ma'am. It was a most honor to interact with you. And thank you so much, Dr. Alexander, for being with us today and providing a glimpse on your talk, which is biomarker discovery and the novel tools to explore the renal pathology. So, folks, mark your calendar for this upcoming talk in the Winicon 2024 to be held on 4th August at 9 to 9:30 a.m. IST at the Hotel Mascot in Thiruvananthapuram, Kerala. We hope to see all of you there and enjoy the session and the conference. Thank you, madam. It's a most honor, and we like to end the interview session. Thank you.